Yeah, good morning. My name is Wei-Ting Chen, and I'm a staff scientist, work with Professor Barty Struper in VIB-KU Leuven in Belgium. And today I'm going to tell our story that we use spatial transformic and in situ sequencing to study Alzheimer's disease. And this story has been published on Cell last year. And the cover story that we want to send a message that amyloid plaque, like the white stuff here, that have a ripple effect throughout the brain cells and to propagate a pathogenic signaling. And by the spatial transformic, it's the first time that enable us to unbiasedly identify what's the molecular mechanism represent this pathogenic signaling. And in situ sequencing that help us to identify what's the cell type to propagate this pathogenic signaling. So, and our hypothesis is we consider Ebitda as the trigger, but might not be eventually the driver of the disease progression. And we learn from the clinical trial anti-amyloid therapy that can actually remove amyloid plaque in human brain, but they don't have significant benefit on the cognitive impairment. Our hypothesis is that once the plaque is there and to the end stage of the disease, at some time point, the plaque will induce a multicellular pathogenic network. And this pathogenic network that might become independent of the presence of the plaque. And if we remove the plaque at the end stage, the cells are still sitting there and keep driving the disease progression. If that is the case, that would be very interesting for us to understand what's the mechanism shared by multiple cell types which drive the disease progression. To allow us to do this, we need the technology with global transformic profile and the spatial information. So the traditional way we look into spatial information is like immunohistochemistry. And we start our study from the mouse model APP NLGF knocking mouse. So here you see the four corner section from 3 to 18 months of age. And the zooming, you can already see the whitish amyloid plaque at a very early stage that can already induce a specific GFAP positive astrocyte, like an envelope around the plaque, which is specific to a particular microenvironment, but not really far from the plaque. And this cellular response is getting severe during the disease progression together with the amyloid plaque. However, if we want to unbiasedly identify what's the molecule and cells involving this plaque-induced alteration, then we need global transformic profile with spatial resolution. We got an early access with Professor Joachim Lemberg in 2016, which is kind of a 2D RNA sequencing array. And on top of the array, they have 1,007 spots. And within each spot, they have more than 200 million oligos. All the oligos, they will have poly T tails. The oligo within the same spot will have the same ID barcodes. And then we do the cryosection to put the frozen brain on top of the array. After fixation and permeabilization, the mRNA will leak out from the cell and attach to the poly T tail. We do the in situ RT and collect all the cDNA afterwards. All the cDNA collect into one tube for further library construction and sequencing. The molecule will bring back to their spatial localization based on the ID barcode. So by this way, then we can collect more than around like 500 to 600 of the transformic profile of each tissue domain in parallel. The spatial resolution of each tissue domain is 10 micrometer in thickness, 100 micrometer in diameter, which is roughly the size of amyloid plaque. The amyloid plaque is roughly in 5 to 50 micrometer in diameter, so they can capture some cells around the plaque. By this way, we collect more than 10,000 of transformic profile across 2 genotype and 4 time points. The Disney plot shown here that shows that the transformic profile can be segregated either by the age or a genotype or the brain regions. They can even identify the microenvironments such as the red curve, the somatic layer of the hippocampus, to differentiate CA1, CA3, and the dentate gyrus. So which give us confidence that it's biological meaningful technology, as long as the microenvironment they have, although that is very close, as long as they have distinguishable transformic profile that can be distinguished by the technology. The next is we want to link those transformic profile to the Ebitda accumulation. So we do the immunohistochemistry and we quantify by image analysis. For each tissue domain, we know how much level of Ebitda is accumulated within each tissue domain. Then for each tissue domain, we not only have the full global transformic profile, but also have corresponding Ebitda index. Then we want to see what's the genes, their expression level is corresponding to the appearance of the Ebitda. And we do it by a totally unbiased way. We use a WGCNA algorithm, which can identify the network, their co-expression together across the database. So we do it across our more than 10,000 of the transforming profile, identify totally 12 networks. So the networks can be driven by the age, by the genotype, and most importantly, our database have spatial information, so they can be driven by brain region or the various level of Ebitda deposition in each microenvironment. And we plot those network, their expression log for change according to the genotype or according to the level of Ebitda. And we plot their log for change in the early stage at 3 months or at 18 months. So the most outspoken network shown here, that is upregulated in AD versus wild type, and also in tissue domain, particularly enriched with Ebitda deposition at 18 months of age, and which is mildly response at 3 months of age. So we call further characterization, we call this network the plaque-induced genes, the PIGS network, which is composed by 57 of the genes. And I show you the detail of the gene list here. So this is Zucker's plot. You can see the green curve, which indicate the correlation of the co-expression of the gene pairs, the strength of their conductivity. The darker the color, the higher correlation of the co-expression of the gene pairs among the selected transformic profile. So here, we only select the transformic profile from the wild type mouse brain, whereas none or very few Ebitda appear. And you see that the network that's split into 3 subgroups. They don't really connect it. So the green subgroup, they have strong internal conductivity, and most of them are seems to be expressed by astrocytes. And the blue subgroup, they have mild internal conductivity, which might be expressed by microglia. So when we select the tissue domain or transformic profile from AD mouse brain, where it have lowest Ebitda deposition, you see the conductivity start to be increased, not only internal, but also external of the subgroup. So the network gradually to be built up. And that is according to the level of the Ebitda deposition. So the connection is strongest in the tissue domain with highest Ebitda deposition. We normally call it amyloid plaque. And the network is especially connected by TREM2, TYRO-BP, ApoE, C1q, ABC. So it's really to our surprise is this totally unbiased analysis, which fish out those 57 of the genes from totally more than 20,000 of the genes. That show us this network is particularly co-expression with the presence of the Ebitda deposition. And the major connector is by the AD risk genes like ApoE and TREM2. So for further gene ontology analysis that indicate the network indicate the dysregulation of classical complement cascade, lysosomal dysfunction, antigen processing and presentation, and the oxidative stress. So the network not only tells us the hint about which target can be further developed for the therapy, They also tell us what's the aspect of the target we should focus. For example, ApoE that have multiple functions in the brain, but here they indicate their connectivity or their interaction with TREM2 and the C1q ABC might be important. Interesting paper published last year in Nature Medicine, which indicate that the ApoE have physical interaction with C1q to inhibit the classical component cascade. So spatial transformic that tell us the novel network and the targets we should focus. However, they don't have single cell resolution. So we incorporate the other technology. We want to know what's the cell express the target that will help us for further target validation and experiments. So the in situ sequencing on the collaboration with Katana that allow us to get the pixel resolution of the gene expression that can detect more than 100 of the genes on top of one section. So the image showing here is the detection of the PROX1, which is a landmark of the dentate gyrus. And the red spot I'm showing here is the detection of the neuron D6 or synaphysin, which are neuron markers. The blue for oligodendrocyte, green for astrocyte, yellow for microglia. So only based on the detection of those genes that already nicely delineate the structure of the mouse brain. And moreover, we can do the immunohistochemistry on top of the same section. So it's really nice to see that the white amyloid plaque is already colocalized with green and the yellow indicate the gliosis around the amyloid plaque. So by this practice, combined with the 57 of the peaks, and then we can understand what the cell type express each peaks and whether that is close or far away from the plaque. And we do this practice on 2 wild type and 2 AD mouse brain at 18 months of age, and also partially validate the result on the human brain. And we are very happy to see that the orthogonal technology that give us a highly correlated result. So this is the level change of the gene expression to Ebitda detected by spatial transformic in the y-axis. And the x-axis indicate the log odds ratio of the gene and the presence in the, around the amyloid plaque. So they both show that two technology that shows that they are all highly enriched around the plaque, especially TREM2 and ThyroBP has the highest enrichment. And furthermore, in situ sequencing that tell us what the cell type express those genes in different conditions. For example, in a wild type mouse, AD mouse, or a particular microenvironment around amyloid plaque. So that tell us that the green cluster that is mostly expressed by astrocyte, the blue and the orange is mostly expressed by microglia. And sometimes the neuron oligodendrocyte also involved in the expression of the network. It's interesting to see that the ApoE is normally expressed by astrocyte and the wild type and AD brain, but they start to be expressed by microglia in a microenvironment around the plaque. And for C4A and B is normally expressed by astrocyte in wild type, but they start to be expressed by oligodendrocyte in AD mouse brain, and even more around the amyloid plaque. So those indicate that amyloid stress, particularly amyloid plaque, that will induce gliosis or further activation and gradually build up this kind of PIX network by the expression of the ApoE in microglia and C4A and B and oligodendrocyte. So that further tells us what's the molecule in which cell type we can further do the target validation. So I hope that until now, I convinced you that new technologies such as spatial transformic and in situ sequencing, they can help us to answer new type of questions, such as uncover coordinated cellular response to amyloid plaque in Alzheimer's disease. So before I move on to our current progress, I would like to highlight that the technology that revoluted extremely fast during the past 3 to 5 years. So here I try to summarize the technology based on their spatial resolution or the throughput. And beyond those two properties of the technologies, I will also summarize what kind of other properties that might be important for the biological application. Sorry. So our paper basically already tells us that those new technology is very interesting. They can help us to answer a new type of question that cannot be solved by traditional technology. And that basically coming from two aspects. The first one, we want to get a huge amount of transformic profile on a lot of separated tissue domain, and the global transformic profile that can help us to unbiasedly identify a network or some targets particularly correlated to a particular small microenvironment such as neuron pathologies. And after we identify such a network, then we want to increase the capability to multiplex the RNA detection in pixel resolution. That will help us to understand the complexity of the cellular response to such a microenvironment of the neuron pathogenic hallmark. However, recently, there are a lot of new technology coming in, either like a 2D RNA sequencing array to increase the spatial resolution, or there are more options that help us to multiplex RNA detection in pixel resolution and to acquire this information by microscope. So when we look at the technology, I not only look at their throughput or spatial resolution, I'm particularly interested about their sensitivity, because that's largely decide to what extent we can identify a mild alteration induced pathology such as amyloid plaque or tau. So I take the reference of the single nuclear RNA sequencing, which is around 10% of the SM fish. That's already almost up to the limitation that we can get the AD-induced robust alteration. So normally I interest on the technology which is higher than 10% of the SM fish. And some technology, although they at the moment don't have a very high throughput capability due to some feasible reason, but they actually provide much higher sensitivity. For example, like 40 to 60% of the SM fish, and that will be very interesting for biological application. And for the other part, I also interested about whether the technology is easy to get hands-on and implement in the lab or an institute for further 3 to 5 year investment. So some of the technology at the moment is very limited in a very limited lab around the world. That might not be easy at the moment to be implemented. So we will also take care about whether that is feasible for further implementation in the institute. And the next, because we work on the proteopathy, so there are a lot of information cannot be acquired by multiplex RNA detection in situ. We need the detection of the protein, such as immunostaining. And here I show you the technology that help us to multiplex the protein detection in situ. And for example, we were interested on whether the protein aggregation is happen in certain microenvironment, like amyloid plaque, or whether there's a phosphorylation of the protein, like tau, or some kinase involved in the necroptosis, or granular vascular degeneration. And furthermore, the immunohistochemistry that can further help us to identify, to realize the morphology of the cell, whether that is hypertrophy of the glia, dystrophic neuroni, and to count the number of the synapse. So if we can combine the RNA and protein detection in pixel resolution on one section, that will be fantastic, and that will tremendously help us to answer many new type of the questions. At the moment, that might be difficult for the commercialized technology to help us from this kind of application. And we also know that the frozen tissue that might help us to maintain the RNA quality. However, that will compromise the morphology of the cells and also limit the capability of the antibody to recognize the epitope. So if we want to combine a protein standing on one section, normally FFPE sample will have higher performance to get a good immunostanding of the protein. So that depends on the question whether we focus on the protein or the RNA detection in situ, then we choose the technology and the sample to apply to answer our particular questions. So here I segregate different technology into 3 groups. Those are the technology only focus on the multiplexing RNA detection in situ. And since I have aware data technology with that share the similar rationale, normally they will have similar capability regarding those 5 aspects. And today we might not be able to mention too much about the detail of the rationale, but those technology that's all highly possible to directly carry out on the frozen on the human material. And that's really the next step that we really want to move on. Because the mouse model, they cannot fully capture AD phenotype. For example, there's no much neurodegeneration, such as tau pathology, necroptosis, or granular vascular degeneration in the amyloid mouse model. And at least 1/3 of AD risk genes, they don't have good mouse orthologs. So I think that is not that surprise if we see the efficacy. They show the benefit in the mouse model to improve the cognitive function, but they cannot really recapture the same efficacy in the human study in clinical trial. So we really want to bring this discovery biology for target mining and target validation in all human-based material. And recently we have some collaboration going on, and hopefully we will be able to update our recent result very soon. So I particularly want to appreciate a lot of colleagues that we work together for our cell paper. In our cell paper, the data analysis is majorly carried out by Ashley and Mark. And we work with a lot of colleagues within Bar's lab in VIB-KU Leuven, especially Kathleen, involving a lot of experimental processing. And image analysis is a lot of involvement and engagement from the VIB image core with the colleagues here. We have collaboration for spatial transformic with the lab from Professor Joachim Lambert. In situ sequencing is on the collaboration with Katana. So both spatial transformic and Katana are now part of Transcriptomics. And the mouse are from Professor Sedo in Riken, and the mouse sample, human sample are from Professor Ingen from Neiland Brain Banks. And from now on, I will start to take the questions.
Wei-Ting, that was fantastic, really clear, and yeah, perfectly to time. So thank you ever so much. And I particularly, not only was the biology really impressive, but I was very grateful for the overview you presented of the different technologies and the platforms and the strengths and how they can help us with the resolution, both the genes and the spatial aspects. That was really great. Thank you so much. It's always a bit flat on Zoom because no one can clap, but ultimately there'll be lots of people clapping. So we've got lots of questions. The first one is one right off my heart from Axel Montaigne. He writes, great talk, thanks. Did you and can you look at vascular cells, so these endothelial cells, parasites, smooth muscle cells in your AD models? And for those of you who don't know, it's very difficult to look at the vascular cells. They're very difficult to free. They're captured in a lot of extracellular matrix. So looking at the vascular component in these single cell approaches is often really underrepresented. So Wei-Ting, can you see these vascular components in your spatial transcriptomics?
Yes, thank you. I think that's a fantastic question. That vascular is particularly hard to be analyzed by single cell and single nuclear approach. And I also think that will probably a niche for the spatial related technology. And however, for all the spatial analysis, that will highly depends on whether we have the capability to do the image analysis and then to identify a particular images or pixels, whether they belong to the blood vessels or the vessels. Yeah. So for the most part, that we did not put that much of the effort to do the image annotation on where is the localization of the vessels and what's the corresponding transformic profile. But that's definitely our next focus. As you can see, like here, we focus on the human brain, and that is human brain section. We put on the 2D RNA sequencing array, and we do the immunohistochemistry before we do the RT. So we will get the RNA and protein detection exactly on the same section. So here you see there are already some small spots, that is the blood vessels. And now we have already put our effort of the image analysis and annotation to further point out which is the potential related transformic profile correlated to the vessels. And we look forward for further analysis. Yes.
Brilliant, brilliant. Thank you. I think it's still going to be tricky, because ultimately, whether you're doing in situ detection or in situ capture, actually either getting the probe into there, I can imagine being harder than the parenchyma, and also, you know, digesting that to release the RNA. So I think that's going to be an interesting frontier. Thank you. So we have another question from Bassem Hassan, who again writes, "Hello, impressive work. If I understood correctly, the hypothesis is that the interesting changes in gene expression in different cell types occur during the cellular phase of the disease, which precedes the phenotypic phase. If so, would it not be more interesting to focus on the changes that occur well before 18 months? So looking looking at these models at a younger time point." He writes, "It seems to me by focusing on the cell gene network at 18 months, you end up discovering the network that responds to the pathology, not the one that creates or propagates it." Do you have any thoughts about that? Have you looked earlier?
Yes, thank you. Sorry. Let me find it. I think my voice today is horrible.
Oh, you're coming across very clear, don't worry.
Yeah. So here we try to provide a proof of concept about whether the technology can provide a meaningful network. So we focus on the network, which is highly correlated with the presence of the Ebitda. But we do also find the other network which is particularly abnormal in the early stage. For example, like this red network, which is upregulated in the 3 months, but downregulated at 18 months. And that is the network highly represented with the oligodendrocyte-related transcript, and of course, some myelination-related processing. And we further check about the detail on the network. So that's actually enriched also with a lot of genes that identify in many other single nucleotide paper from the frozen human brain that identify the disease-associated oligodendrocyte that's also enriched within this network. So we found that probably our mouse model that represent in a very early stage, maybe there's some rebounds of the myelination around the amyloid plaque, but eventually they cannot hold, and eventually there's a downregulated in the very intensive Ebitda deposition around the amyloid plaque. But that's at the moment, this is more a complicated question that might need further investigation by the other target validation. But we also aware that the oligodendrocyte is a very interesting alteration in the Alzheimer's disease. For example, like OPC, the oligodendrocyte precursor cells that might undergoing senescence in the amyloid plaque, so they cannot become mature to have proper function of the myelination in the high intensive amyloid plaque depositions. So that might a particular alteration around the amyloid plaque. But for here, we cannot say that much at the moment. Yeah.
Right. So interesting that ODCs, oligonucleotides are coming through. But that also leads me on to sort of the question about genetic risk. So when we look at Alzheimer's, the genetics has really guided us towards microglia. So we see microglia as perhaps the instigating cell type, whether that's true or not, whether that's a simplification, I don't know. But in your WGCNA networks, in your 10 micrometer sections, you see both microglia and astrocyte networks, at least dysfunctional networks, which occur, you think, in astrocytes and microglia. Are you able to see which comes first? Are you able to see instances where you see perhaps the microglia behaving aberrantly, but not yet in the astrocytes, or vice versa? So you're able to give an idea of the ordering in which these cell types are recruited to the pathological process?
I think that's definitely a fantastic question that I did not think in depth. So if a quick question coming from my end is probably like this one, because we feel like this network, it seems like this is an astrocyte subgroup, and this is probably mainly leached by the microglia. So it seems the network that have, there's no much communication between those two subgroups in the wild type, but they start to be built up in the mild Ebitda deposition. And so which come in first, or which come, maybe, I don't know, maybe they have more internal conductivity and higher expression of the astrocyte subgroup in our PIX network in the wild type brands. But when we start to have more Ebitda deposition, there seems to have more interaction, but what aspect of the interaction that we might further get some hints from the connectivity between the gene pairs. But I think that's definitely a fantastic question. I need to think more about it.
Good, good. What we're here to do. So then one last question from Blanca. And Blanca writes, hi Wei-Ting, beautiful presentation. I noticed there's some astrocyte and microglia genes whose expression is reduced around the plaques. Do you know what kind of functions they perform?
Sorry, I'm not sure whether I got a question. So
Blanca notes that that there was some astrocyte and microglia genes whose expression was reduced. So these are genes which were downregulated around the plaques. So specifically the genes that were downregulated, what kind of functions were they involved in?
Okay, so from our mouse model, it seems most of them are upregulated around the plaque. Yes, so regarding the function
of the downregulated ones, if it's clear.
Yeah, so here it seems they are mostly all upregulated.
Right.
But for the human brain, that's only like 14 to 16 of the gene is upregulated, and the other we cannot show the replicated SAM in the human brain. Yeah.
Brilliant. Well, thank you very much. That's a really clear and excellent and provoking talk. So thank you very much for that. So if you can stop sharing, then we will move on to the next speaker. Thank you.
Thank you.
Great questions. Keep them coming.